I frequently think of Thousand Genomes as a Lewis and Clark expedition to the interior of the continent. We knew the outlines and the contours. We understood where the major rivers and the bays were.
A team of researchers in China quietly built one of the largest genetic studies ever done on a single population, and the numbers they pulled out of that data did not match anything the textbooks had been teaching for years. The findings traveled through hospitals, drug companies, and government agencies in ways most people never noticed. What they actually found is more useful, more surprising, and far more important than the version that usually gets told online. The study that was never supposed to be controversial. Before getting into the older history, here is where things stand right now. As of late 2025, the United States government signed something called the BioSecure Act into law.
US, which has gone ahead and passed the US BioSecure Act
As part of the yearly defense spending package. This law restricts federal funding from going to certain Chinese biotech companies, and the Beijing Genomics Institute, often called BGI, sits right at the top of that list. Earlier in 2023, several BGI branches were already added to the United States Entity List, which is a kind of trade restriction list, after concerns about gene data being shared with the Chinese military. So, the company at the center of this story, it has become one of the most watched biotech firms on the planet. Now go back to October 4th, 2018. The lead authors were Saiyang Liu, Shujia Huang, and Fang Chen, working with a team that included Rasmus Nielsen from the University of California, Berkeley, and several senior researchers from BGI. The full title of the paper discussed non-invasive prenatal testing, viral infections, and the history of the Chinese population. The study did not sequence 140,000 full genomes. What the team actually did was use leftover blood samples from 141,431 Chinese pregnant women who had taken a routine prenatal test. That test reads only a small slice of the genome, somewhere between 6 and 10% per person. The team then used a clever statistical method called imputation to fill in the gaps. So the right way to describe the work is that they used ultra-low coverage data from a very large number of people and stitched it together with the help of a smaller, deeply sequenced reference group of 40 individuals. The original goal was very practical. The team wanted to map the genetic structure of the Chinese population, find new disease links, and study viral patterns in pregnant women. The participants came from 31 of the 34 administrative regions of China. There is one more myth worth clearing up. Some online versions claim that peer reviewers fought to block the study or rejected it 3 times because the findings were dangerous. There is no public record of that happening. The controversy that came later was about something completely different, and it had nothing to do with whether the science was sound. That second wave of attention started in July of 2021, when a Reuters investigation showed that BGI had been working with Chinese military hospitals and that gene data from millions of women across more than 50 countries was being stored on Chinese servers. That investigation, not the original Cell paper, is what put BGI on the radar of governments in Washington, London, and Brussels. The science the team published in 2018 and the geopolitical fight that followed are 2 different stories. They are often blended together online, and that mix-up has done a lot to confuse what the study actually showed. So, with the dates fixed, the methods clarified, and the corporate background in place, the next thing to look at is what the team found inside the data itself. Because the moment they started comparing northern Chinese samples with southern Chinese samples, the lines on their charts started behaving in ways the team had to explain very carefully. When genetic diversity stopped making sense. The first surprise inside the data was about how the Chinese populations divided up internally. In population genetics, scientists use a measure called FST to describe how genetically different 2 groups are. Higher numbers mean more difference. Using widely cited values from the 1000 Genomes Project, the FST between Han Chinese from Beijing and people of European ancestry from Utah sits around 0.10. Within East Asia, the FST between Northern Han and Southern Han is much smaller, but it is real, and it follows a clear north-to-south line that runs along a natural boundary called the Qinling-Huaihe Line. The team found something else that was unexpected. Going east to west across China, the genetic differences were very small. The paper itself noted that this was perhaps surprising. One likely reason is that large internal migration programs after 1949 moved many people across the country, which mixed regional groups together and softened older boundaries. Another finding is tied directly to history. People who self-identified as Manchu were genetically almost identical to Northern Han populations, which lines up with centuries of Sinicization. People who self-identified as Hui, the Chinese Muslim community, carried about 5 to 10% West Eurasian ancestry, likely a trace of Silk Road trade and migration over many generations. Then came one of the most interesting genetic differences between East Asians and Europeans. There is a gene called EDAR, which stands for ectodisplacin A receptor. The specific variant of interest is correctly named V370A. Many online videos call it V378A, but that is a misprint that has spread without correction. The technical reference number is RS3827760. This variant is present in around 94% of Han Chinese individuals tested, around 87% of Koreans, and around 78% of Japanese. In modern Europeans, it is essentially absent, which means close to 0%. The figure of 5% that often gets quoted is wrong. There is a separate East Asian variant called RS146567337 that does sit around 5% in some southern Chinese and Southeast Asian populations, and the two variants are likely being mixed up by people repeating the story online. A separate research group led by Leslie Hlusko at the University of California, Berkeley, pushed the story further. Their 2018 paper in the Proceedings of the National Academy of Sciences proposed that the variant was strongly favored during a long stay of human ancestors in the Bering region between Siberia and Alaska, possibly because the changes in mammary gland branching helped infants get more nutrients during long, dark, vitamin D-poor winters. Hlusko explained in interviews that high-latitude populations would have been short on vitamin D, and that the modified breast tissue may have shifted the fatty acid balance of breast milk in a way that helped babies survive. This is one working hypothesis among several, and the field still has open questions about exactly which traits drove selection. The point is that EDAR alone is not just a hair and teeth story. It connects to skin, sweat, infant feeding, and possibly the entire migration story of how people moved from Asia into the Americas. Once researchers understood how deeply this one gene was woven into so many systems, the next question became obvious. If one gene could carry that much history, what would happen when the team looked at the wider catalog of genes that had been pushed up in frequency by recent natural selection? That is where the next layer of findings starts to open up. The selective sweeps. Nobody predicted. The phrase often used here is selective sweep, which means a useful gene variant rises quickly in a population because it gives a real survival or reproductive edge. East Asian populations carry several of the most studied selective sweeps in modern human genetics, and most of them connect to something very old and very ordinary, which is food. The first example is a gene called ADH1B. This gene helps the body break down alcohol. A variant called ARG47-His, identified by Bing Su and his team in a 2010 paper in the journal BMC Evolutionary Biology, sits at over 50% frequency in 31 of 38 East Asian populations sampled. In some Chinese groups, it climbs above 98%. In Europeans, the same variant is found in only about 2 to 10% of people. This variant speeds up alcohol breakdown by a factor of 40 to 100. Bing Su and his team estimated the variant became common in East Asia somewhere between 7,000 and 10,000 years ago. That timing matches almost exactly the rise of rice farming in southern China, which is also when fermented rice-based drinks became part of daily life. The next gene is called ALDH2. The famous variant here, named RS671, is the reason many East Asians get a flushed red face after drinking alcohol. Around 30 to 50% of East Asians carry at least one copy of this variant, while it is essentially absent in Europeans and Africans. The combined effect of these two genes means that the body of someone carrying both variants converts alcohol into a toxic byproduct very quickly and then struggles to clear it. This protects against heavy drinking, but increases the risk of certain cancers if a person drinks regularly anyway. Then there is the FADE gene cluster, which controls how the body makes long-chain fatty acids out of plant or animal fats. Work by Ian Mathieson and others using ancient DNA showed that European farmers were selected for one version of these genes that helps process plant-based diets, while Greenlandic Inuit were selected for the opposite version, suited to a diet heavy in seal and fish fat. Matteo Fumagalli, a co-author on the Inuit study, said in press coverage that it was fascinating how Greenlanders ended up with a unique genetic makeup that let them better use their traditional food sources. The bigger point is that the same gene region was pushed in opposite directions in different populations because they were eating completely different things. European populations went through their own well-documented sweeps. The most famous involves a gene called LC, which controls the body's ability to digest milk sugar in adulthood. Joel Hirschhorn and his colleagues showed in 2004 that this variant rose dramatically in the past 5,000 to 10,000 years, lining up with the spread of dairy farming. What ties all of this together is timing. Almost every one of these strong sweeps happened in the last 10,000 to 30,000 years, well after the first migrations out of Africa. That means these were not ancient inheritances. These were recent adaptations to specific places, specific foods, and specific climates. The hard sweep model of one mutation racing to fixation has also been challenged by the work of Jonathan Pritchard and his colleagues, who proposed that much of human adaptation happens in a softer, more spread-out way across many genes at once. All of these signals are real, well-measured, and largely settled. But there is one part of the modern human genome that is far stranger than even these sweeps, because some of it does not appear to come from modern humans at all. The ghost DNA problem. Inside the genome of nearly every person alive today are small fragments of DNA inherited from human relatives who went extinct tens of thousands of years ago. Europeans, on average, carry about 1.5% to 2% Neanderthal DNA. Mainland East Asians carry slightly more Neanderthal DNA, around 1.7 to 2.1%, plus a smaller amount, around 0.2%, from another extinct group called the Denisovans. People in Papua New Guinea and parts of the Philippines carry the highest Denisovan ancestry on Earth, in some cases between 3 and 6%. The Eta people in the Philippines reach around 5% Denisovan ancestry, the highest reliably measured anywhere. The most famous example of how this archaic DNA still matters today is the Tibetan altitude adaptation story. Rasmus Nielsen, working with BGY and his Berkeley team, published a paper in the journal Nature in 2014 showing that a gene called EPAS1, which helps the body manage low oxygen, was inherited in a special form from Denisovans and is now found in about 87% of Tibetans. Only about 9% of Han Chinese carry it. In an interview with Berkeley Press, Nielsen said that part of the Tibetan EPAS1 gene is almost identical to the gene seen in Denisovan remains, and very different from all other modern humans. He said the only real explanation is that this stretch of DNA was inherited from interbreeding with Denisovans, and that there was no other way to explain the data. He went on to say that this is one of the clearest examples of humans evolving and adapting to a new environment by getting useful genes from another species. There may still be more interbreeding events left to discover. Sharon Browning at the University of Washington, in a paper in the journal Cell in 2018, showed that there were at least 2 separate pulses of Denisovan mixing into the ancestors of modern humans. In her own words, she said that DNA analysis had revealed there were at least 3 interbreeding events in our deep past, with at least 1 Neanderthal pulse and at least 2 Denisovan pulses. Murray Cox and his colleagues later found evidence that the center of archaic human diversity was probably not in icy northern lands, as people once assumed, but in tropical Asia. So, the human genome, as it sits inside every person reading this script, is not a single neat blueprint. It is a layered record of many encounters. Some of those encounters left useful gifts, like the Tibetan altitude gene. Others left puzzles that scientists are still trying to solve. The reason these archaic fragments matter is not just for storytelling. They actually change how the body works. And once researchers started lining up these differences with real medical data, they ran into a problem that hospitals and drug companies had been quietly ignoring for years. The medicine built on the wrong population. For decades, most large genetic studies, drug trials, and disease risk calculators were built almost entirely on people of European ancestry. The numbers here are well documented. In 2016, Alice Popejoy and Stephanie Fullerton published a piece in Nature called Genomics is Failing on Diversity, which showed that about 81% of all participants in genome-wide studies were of European background. By 2024, despite years of public commitments to fix this gap, the cumulative figure had actually grown worse, sitting around 94% European ancestry. The main reason is that the United Kingdom Biobank, which is mostly white British, has come to dominate the field. This imbalance has very real medical consequences. Take type 2 diabetes. The strongest common gene variant linked to type 2 diabetes risk in Europeans is in a gene called TCF7L2. The risky version is found in about 25 to 30% of Europeans, but only 3 to 5% of East Asians. The biological effect of the variant looks similar across populations, but because it is much rarer in East Asia, it explains far less of the disease burden there. A different gene called KCNQ1 was almost completely missed by early European studies, because the relevant variant was uncommon in Europeans. 2 Japanese teams, led by Kazuaki Yasuda and Kazuo Unoki, published nearly simultaneous papers in Nature Genetics in September of 2008, showing that this gene was a major diabetes risk factor in East Asians.
Experts say genes, not just diet, can drive the health of our gut microbiome and protect us against diabetes.
Around 40% of East Asians carry the higher risk version, compared to about 7% of Europeans. If those Japanese researchers had not run their own studies, the gene would have remained invisible. Drug response is even more striking. The blood thinner warfarin works through a gene called VKORC1. The high-sensitivity version is found in close to 90% of East Asians, but only 40% of Europeans. As a result, East Asian patients typically need warfarin doses around 30 to 50% lower than European patients. Giving the standard European dose to an East Asian patient can cause dangerous bleeding. Another example is a heart medication called clopidogrel. A variant in the CYP2C19 gene reduces how well the body activates the drug. Around 55 to 65% of East Asians carry at least one copy of this variant, compared to about 30% of Europeans. A landmark study led by Jessica Mega at Brigham and Women's Hospital in 2009 showed that carriers had a 53% higher rate of major heart problems after stent placement. Then there is carbamazepine, a common epilepsy and nerve pain medicine. A team led by Wen Hung Chung in Taiwan published a study in Nature in 2004 showing that a tissue marker called CHEL, AB1502, common in many Han Chinese populations but rare in Europeans, was strongly tied to a life-threatening skin reaction in patients given the drug. In their original sample, every single case carried this marker. The Food and Drug Administration in the United States added a boxed warning in 2007 recommending genetic testing in Asian ancestry patients before prescribing. Polygenic risk scores, which try to predict disease risk from many genes at once, are also far less accurate outside of European populations. Alicia Martin at the Broad Institute showed in a 2019 paper in Nature Genetics that current scores can be up to 4.5 times less accurate in people of African ancestry than in people of European ancestry. In her words, this disparity is a direct consequence of how skewed genome-wide studies have been, and clinical use of these tools today would systematically benefit European descent populations much more than anyone else. And when this fight first started getting public attention, it ran into a much older argument that the field had been trying to leave behind for decades: the fight to publish. The internal structure of the Chinese population that the BGI team described in 2018 has now been confirmed by 3 separate large-scale projects. A team led by Yanan Chao published the China MAP study in the journal Cell Research in 2020, using more than 10,000 deeply sequenced genomes. Another project called NUA, led by Peng Zhang and colleagues, came out in Cell Reports in 2021. A third effort, the Westlake Biobank for Chinese, led by Peng Yu Kong, was published in Nature Communications in 2022. All 3 independently found the same north to south structure along the Qinling-Huaihe boundary, the same softer east to west split, and similar selection signals at ADH1A, ADH1B, and ALDH2. Outside of China, 2 large biobanks have made East Asian human genetics far more visible. Biobank Japan, led by researchers including Yoichiro Kamatani, has now combined with the United Kingdom Biobank and the Finnish FinGen project to study more than 600,000 people across many traits. The Korean Genome Project has done the same for Korean populations.
Now, the age of gene therapy is fast approaching. Doctors first started trying to heal genetic disorders back in 1990.
These projects have repeatedly confirmed that gene variants found in East Asians often work differently from the way they do in Europeans, and that some risk factors only show up clearly when East Asian samples are studied directly. Diet adaptations show a similar pattern. A study led by George Perry, then at Arizona State University, showed in 2017 that populations with high-starch diets, including the Japanese, average more copies of a gene called AMY1, which makes a saliva enzyme that breaks down starch. Populations whose ancestors ate fewer starches average fewer copies. Later work by Charles Lee and his colleagues refined the measurement methods, but the basic story has held up. Europeans went the opposite direction with milk, where lactase persistence rose because of dairy farming. Even within the past few years, work led by Richard Evershed at the University of Bristol, using ancient pottery residues, has shown that European populations were drinking milk for thousands of years before the gene. For adult milk digestion became common, which means the selection pressure that finally made the gene widespread is still being debated. Microbiome research has added one more layer. Tanya Yatsunenko and her colleagues showed in a 2012 Nature paper that the gut bacteria of people in the United States are very different from those of people in rural Venezuela or Malawi, and that these differences appear from infancy. A later study from Huijue Jiang and her team in Nature Medicine in 2018 looked at more than 7,000 people in just one Chinese province, Guangdong, and found that microbiome-based health prediction models built in one part of the province failed when used in a different part. The conclusion is that genetics, diet, and bacteria all interact in patterns that can be very local, not just regional, But there is one part of the field where this distinction has been ignored over and over, and where some of the loudest claims have collapsed under careful testing. The genes nobody wants to talk about, and what comes next. In the mid-2005s, 2 papers in the journal Science set off a long argument. A team led by Bruce Lahn at the University of Chicago reported that 2 genes involved in brain development, called ASPM and MCPH1, showed signs of recent natural selection, and that the selected versions were more common in some populations than others. The papers carefully avoided making direct claims about cognitive ability, but the implications were obvious to everyone who read them. What followed was one of the cleanest examples in modern genetics of how a popular hypothesis can fall apart under scrutiny. In 2006, Matthias Kuret and his colleagues showed that the same statistical patterns Lahn's team had highlighted could appear without natural selection at all, just from population history. In 2007, Lahn's own team published a follow-up paper using close to 2,400 subjects across 5 samples. In their own words, the overall findings did not support a detectable association between the recent evolution of these genes and changes in measured intelligence. Studies by C. Jeffrey Woods, Nicholas Timpson, and others using brain scans and large child cohorts came to the same conclusion. Even Philippe Rushton, who had spent his career looking for genetic links to cognitive differences, could not find one in this data. The same fate met another famous gene called DRD4, which has a variant sometimes nicknamed the Wanderlust gene. Frequency does vary across populations, but careful studies by Mihai Naka and others showed that this variation looks like normal random differences, not strong selection. Marcus Munifo led a meta-analysis in 2008 that combined 36 studies of novelty seeking and found no clear effect. Dan Eisenberg, working with Arial pastoralists in Kenya, showed that the same variant was linked to better nutrition in nomadic men, but worse nutrition in recently settled men. In other words, the gene's effect depended entirely on the environment. So the consensus answer to the question of whether genes like ASPM, MCPH1, and DRD4 explain cognitive differences between populations is a clear no, supported by the very researchers whose early work first raised the question. This is not a sensitive area being avoided. It is a settled area where the evidence pointed in one direction. There is still real debate about how to talk about all of this in public. David Reich, a Harvard geneticist, wrote a piece in the New York Times in 2018 arguing that scientists need to stop pretending that all average differences between populations are imaginary. 67 scientists, scholars, and ethicists, including Dorothy Roberts and Jonathan Kahn, wrote a public response saying that Reich's framing risked giving cover to ideas that the evidence does not actually support. Both sides agreed on one point. Findings about human variation will be misused if scientists do not explain them clearly, and they will also be misused if scientists try to hide them. The history behind this caution is heavy. The American eugenics movement, led by figures like Charles Davenport and Harry Laughlin in the early 20th century, lobbied for state sterilization laws that affected at least 60,000 people in the United States. The 1927 Supreme Court decision in Buck v. Bell, written by Justice Oliver Wendell Holmes, has never been formally overturned. Laughlin's model laws were studied and adapted by the Nazi government in 1933. Scientific organizations that pushed racial ranking persisted long after the war, and the modern field of population genetics has spent decades trying to make sure that real findings are not packaged as old prejudices in new clothing. Where the field is going next is more hopeful. In 2022, a project called T2TC QTEM13, led by Adam Philippi and Karen Miga, completed the first truly end-to-end human genome, filling in gaps that had been left blank since the original Human Genome Project. In 2023, the Human Pangenome Reference Consortium, led by Wenhua Li and others, published its first draft pangenome in Nature, which uses 47 diverse individuals and 94 full sets of chromosomes to better represent the variety of the human species. The All of Us research program at the United States National Institutes of Health released a dataset in 2024 with 245,388 clinical-grade genomes, of which 77% came from communities historically underrepresented in research and 46% from minority racial and ethnic groups. Eric Green, the director of the National Human Genome Research Institute, has said that researchers and clinicians using genomics need a reference that reflects the real diversity of the human population, so that genomic medicine does not deepen existing health gaps. The honest summary of all this work is simple. Human populations carry meaningful genetic differences, shaped by their environments and their histories. These differences matter for medicine, for understanding our shared past, and for designing better treatments. They do not rank people. They do not predict character or ability. They map adaptation, migration, and food, not worth. That is the real story of what happens when scientists compare Chinese DNA with European DNA and the parts that usually get hidden under the noise. What surprised you the most, and what should we cover next? Drop your thoughts in the comments below.